Hey gang, in this lecture we're looking at the expression of genes, and then we'll also talk about mutations. Um, Gene expression is what we sometimes call the central dogma of biology. In other words, the main idea of biology. The main idea is that you have that living things have a genome, and based upon that genome they can build things. In order for something to stay alive, it has to be able to make products. And the genome is the building instructions for those products. And there has to be some way that the cell can use the information in the genome to actually physically build something. That is what we mean by expression of genes. Even though a chromosome may have many genes on it, they're not always being expressed. Only seldomly will a gene perhaps turn on and then actually build the product that it codes for, and then other times it might turn off. So when a gene is being expressed, that means that the gene is actually being used to build a product that it codes for, and when the gene is not being expressed, it means that the information, while it's still intact, still stored in the nucleus, it's not being used to build anything. Okay, here's what the process looks like. We start off with DNA. Remember, DNA is housed in the nucleus, and it's housed in your genome. The genome, again, think of it being like a library full of building manuals or instructions for how to build something. And much like a library, how all the books aren't always being used, many of them are just sitting on the shelf waiting for the proper time. That's how it is in your genome. You have many genes that are housed here. They're not always being turned on. They're not always being used. Okay, if the cell needs to build something, it has to pinpoint the specific gene of interest and actually make a photocopy of it. And that process in and of itself is an immensely complicated process. You know, just think about that for a second. Like if I was to go into a library and had many books there, and I had to find just a couple pages in one of those books and make a photocopy of it, it would take me some time to figure out where that segment is, right? It would take me some time to figure out what book it is that I'm looking for, and I'd have to find the right page, and I'd have to find the right paragraph. Well, the cell can do this without, a conscious mind, which I always find is fascinating. So there's an intense amount of regulation there governing how the cell can even know that it needs to turn a gene on, and then even know where to find that gene. I mean, that's nothing short of a miracle in my mind. Okay, but in either case, if it knows it has to turn something on and it can actually find it in the genome, it will make a copy of it. And that copy is known as RNA, and the process is known as transcription. So DNA is transcribed into RNA. Now, there are different flavors of RNA. Our RNA is actually used to make ribosomes. So we talked about how ribosomes are the organelle that are the site of protein synthesis, and they are formed from this stuff called rRNA, and that happens here in this darker staining spot of the nucleus, which is called the nucleolus. Okay, tRNA stands for transfer RNA, and this is going to be the main link between nucleic acid and protein. Genes are always used to make proteins, and then the proteins that are built can then go on to help make all the other types of things. You know, remember, there's different types of elements and different things that the cell has. There's carbohydrates, there's lipids, there's nucleic acids, and there's proteins. Well, the genome only codes for the production of different proteins, and then with the help of those proteins, principally these things called enzymes, which help to facilitate chemical reactions, which help to facilitate building and dismantling of products. All the other classes of macromolecules can also be incorporated and used and employed to build a variety of products. Okay, so the genome codes for protein. It doesn't code for lipid products or carbohydrate products generally. And tRNA is a great link because it speaks both languages. It has on it portions of it that are able to bind to amino acids or protein, and the other part can bind to a nucleic acid. So it sort of stands as the link there, and we'll talk more about that later. Okay, the other component of RNA is mRNA, and this is called messenger RNA because this is the one that actually will leave the nucleus and go out to find a ribosome and serve as the building instructions for that protein factory that's either going to be on the rough endoplasmic reticulum. Ribosomes can either be fixed on the rough ER, or they can be free out in the cytosol, and protein can be made at any of them. So the mRNA has to get out of the nucleus and travel to one of those ribosomes, and then there, its information that it has can be used to assemble a protein. This process looks something like this. Okay, here we have a chromosome, and here we have the DNA, and we're sort of showing what a snapshot of the inside of the DNA. Remember, DNA double-stranded. It has a sequence of bases on it, and just like letters and words, that's what creates the information. In the same way, we can rearrange, the letters of our alphabet to make an almost infinite amount of words. So also these 4 letters, A's, C's, T's, and G's, can be rearranged to make almost an infinite combination of building instructions. So let's say a cell needs to turn on a gene or express a gene. What happens first is that the RNA polymerase will get turned on and bind to what's known as a promoter. And promoters can vary. A common feature that we find in them is they have this sequence TA. It's sometimes called a TATA box. But they're generally upstream from the gene of interest, and this is a major site of regulation, because usually what happens is there's a transcriptional complex that's formed there. Different proteins will bind to the promoter, and then that's what signals to the cell it's time for transcription to occur. Remember, if transcription occurs, then the cell is going to start building things, and the cell does not want to start building things if it doesn't need to build them. That can actually be toxic to the cell if it builds too much of something. So the cell has to walk this fine line. It has to build the product when it's needed, but then shut off production when it's not needed. And promoters are a major site of regulation for that process. There's different things that can either bind to turn on or turn off the promoter, and thereby turn on and turn off transcription. But if the promoter is active and the complex has been established, and it recruits the transcribing machine, which is called the RNA polymerase, then the RNA polymerase will bind, and it will literally just go along and read the sequence of bases on the template DNA strand. And it's the job of RNA polymerase is fairly simple. It's just going to read the template strand and add a complementary base on the new strand that it's building. And it's often the case that RNA is only single-stranded, so that's another big difference between it and DNA. DNA is double-stranded usually. RNA is usually single-stranded. It can fold in on itself to do a variety of jobs as well. Okay, so RNA polymerase, this is an enzyme. Enzymes are molecules that end with this suffix here, ase, and it is the RNA polymer ase. So it's the enzyme that binds and makes the RNA polymer. So you can see here it's going along, and so it sees like an A, and so it adds the complement. RNA has not, in DNA we have A's, C's, T's, and G's. In RNA we have A, C's, U's, and G's. So another big difference between DNA and RNA is that it uses the base uracil instead of thymine. So if you're looking at like a strand of nucleic acid here and you see a bunch of U's and not T's, you're looking at a strand of RNA, not DNA. That's one of the really big differences. Okay, so here it's reading, let's say, a G, so it's going to add the complement, which would be C. Here's another G, so it adds another complement C. Here it has an A, and normally if it was DNA it would be T, but in this case it's RNA, so it adds a U. And then here's a T, so it adds the complementary A. So it just goes along and adds a complementary strand as the polymer is built. Once the transcript is made, then it goes out to the ribosomes, and it is actually turned into protein. This process is called translation because it involves, much like converting one language into another language. Here we have two languages, the language of nucleic acid, and then there's also the language of proteins. And there has to be a way to convert from nucleic acid to protein. So we're translating into protein, and that's what's happening at the ribosome. Okay, now the genetic code is what establishes sort of the whole process. So here I'm showing an RNA transcript that's being made, and there are different tRNA molecules out there that have these things called anticodons on them, and based upon the anticodon that they have, they carry a specific amino acid. So transfer RNA molecules are sort of like the link of why this all works, but they sort of determine the genetic code. So we'll see that later in a minute, but for now what you have to understand is that there are three base what are called codons, so on mRNA, and they code for the addition of particular amino acids, which is what these little letters are here. There's lots of different amino acids. They have different chemistry to them. Some are non-polar, some are polar, some are positively charged, some are negatively charged. And remember that a protein is in essence just a growing strand of amino acids. And based upon the sequence of those amino acids, the protein can fold in on itself and have a unique structure, and it's the unique structure that determines its shape. For example, often it's the case that like receptors on the membrane of the cell, they might have regions of the protein that are hydrophobic, and they form what are called transmembrane domains. They form these little sequences of amino acids that are all hydrophobic, and so they can embed themselves into the membrane. And so in those particular proteins, there would be big long sequences here of a whole bunch of amino acids that are non-polar, meaning that they would be hydrophobic. So just an example there. But in either case, when you see these letters, you should be thinking that it's talking about a specific amino acid. So like SER, that stands for serine, which is right here. And when you see LEU, that stands for leucine. And when you see PRO, that stands for proline. Okay, so there are different amino acids that are going to be added in a growing chain, and it is the letters on the transcript that determine the sequence of the amino acids that are added to the polypeptide, and then consequently later which forms later the protein. Okay, so here's my mRNA transcript, and usually the way that it begins is it usually begins with this codon here, AUG. So AUG usually sets what's known as the reading frame. In other words, what am I going off of here? What establishes the frame of the triplets or the codons? Okay, so AUG is what's usually considered the start codon. It codes for the addition of methionine, and then from there the reading frame is established. So now the codons will be the triplets downstream of that. And so like the very next one here, if this is AUG, my start codon, the next codon would be GGC. So if I was curious to know what this next codon in the transcript would code for, I just come over here to use the genetic code. It's like a little decryptor, right? So you hear this says the first letter of the codon, so that would be G. So I know I'm somewhere in this row. And then the next letter is also G in that codon, and so this is the second letter. You'd go to this column. So now I know I'm talking somewhere in this box here. And then the next letter was C, so GGC. GGC codes for the addition of glycine. Okay, this amino acid right here. The next codon here is GUU. So if I wanted to know what amino acid that codon would code for, I could just use the genetic code again, GUU. Codes for valine. Okay, so that's what the code does. It helps us to determine what is going to be the sequence of amino acids based upon the sequence of nucleotides in the RNA transcript. And again, it's that AUG codon that starts the reading frame, meaning what are we supposed to count as the triplets? What are they going to be the codons? And if the reading frame gets shifted, we'll see this later. It is going to greatly alter the protein that would be made, and that does sometimes happen. When a RNA transcript is made, it doesn't just go straight away to the ribosome and get made into protein like I've thus far led you to believe. It has to undergo what's called mRNA processing. So here's sort of an illustration. This is showing an mRNA transcript, which we would call pre-mRNA because it hasn't been processed yet. And there's usually a 5' end and a 3' end, and so you can think about there being a whole bunch of A's and C's and G's and U's in here, okay, the big transcript. The 5' end and the 3' end refers to the direction of the nucleotide. And remember, a nucleotide is a phosphate, a sugar, and a nitrogenous base, and it can either be facing this way or it can be facing this way. And remember, like DNA is anti-parallel, so one strand they are facing this way, the other strand they're facing the other way. Okay, so in either case, there's a 5' end. There's directionality to a transcript. There is what's called a 3' end and a 5' end, so it helps researchers know which direction the transcript is going. There are in an RNA transcript things called exons, and there are also things called introns. Exons, the way I think about it is E stands for expressed. So these are the portions of the transcript which will actually be used to code for production of something. There are also in mRNA these regions called introns, and introns usually get spliced out. So they used to be called junk. The DNA that coded for them was called junk DNA because we were seeing that they just get cut out and they're not even used. But it's been recently, or at least in the past few decades, it's been shown that the introns are actually important. Okay, but those will all get cut out generally. So when the mRNA is finally made, it looks something more like this. And so to make it, these are the different things that happen. RNA is going to coat the pre-RNA with stabilizing proteins, and then the 5' and the 3' ends are coated. So the 5' end gets what's called a 5' cap, and the 3' end gets what's known as the poly A tail. It's just literally a whole bunch of adenines, one after the next, tons of them, and these caps are going to help to protect it as it exports out of the nuclear pore into the cell. And they also are going to be sort of like a tag that the cell uses to determine where this mRNA is going to go. And then they also help to attach to the ribosome when it finally does get to the ribosome. So a pre-mRNA would have a difficult time getting out of the nucleus, and it would also have a difficult time finding its way to a ribosome and attaching to the ribosome, whereas the mature mRNA that has had the caps on it, it can get out of the nucleus and then it can attach to a ribosome. Okay, and then the introns are spliced out. Now there's this phenomenon known as alternative splicing where the arrangement of introns that are spliced out can vary, and it can actually create multiple variants in the transcript so that one gene could actually code for a variety of different products that are similar but not entirely the same. So that can happen, and it's a way that the cell can use one gene to make multiple products, alternative splicing. Okay, so this is mRNA processing, and at the end of the day, the mRNA that is brought to the ribosome will look more like this. It will be mostly just the exons, and it will have the cap and the poly A tail. Okay, once we get to the ribosome, that's where this process known as translation occurs. So here we're seeing an illustration of it. A ribosome looks like this. It has a small part to it, and it has a large part to it, which is called the large subunit. And again, these are going to be made out of rRNA. So that's the purpose of rRNA, to help make the ribosome. Okay, the ribosome has an A site, and it has a P site, and it has an E site. And how it works is that the mRNA is going to bind and then read through the subunits of the subunits of the ribosome. So the small subunit and the large subunit, they sort of clamp. Think of them clamping on to the mRNA transcript. And so this mRNA transcript, this has just come from the nucleus. And remember, it's a photocopy of the genome, and now it's reading through the ribosome almost like a little scanner. Now, in the A site, tRNA molecules can enter. So that's what you're seeing here. These are all tRNA molecules, and what you should know about a tRNA molecule is that it has one part on it which is called an anticodon, and this is what binds to the mRNA. Okay. There's also a region on it that is going to bind to a amino acid. And this is why the genetic code works, because the amino acid that is attached is specific. It's not willy-nilly. There are specific tRNA molecules out there, and they all vary in the anticodon that they have. And in a tRNA molecule with a certain anticodon can only hold on to, or generally only holds on to, a certain kind of amino acid. So then the tRNA molecule becomes the bridge. Every codon leads to the addition of a specific amino acid because the tRNA molecule that binds to the codon carries with it its specific amino acid. When a tRNA molecule is holding on to an amino acid, it's called charged, and as soon as it releases its amino acid, it can then exit out of the E site of the ribosome, and now it is uncharged. It doesn't have the amino acid anymore. So in essence, as the mRNA is passing through, codons are brought into this A site, and that's where the tRNA molecule can bind. There's going to be a lot of tRNA molecules out here, and as soon as the codon enters the A site, whichever tRNA has the corresponding anticodon will be sucked in and bind to it. Okay, and the speed at which this happens is absolutely astounding. So then, the amino acid is going to be brought in, and then in the P site, I think it stands for peptidyl, because it's the place where the peptide bonds can form. The amino acid can form a peptide bond, and now you have it growing onto the chain. So there's a new protein that's emerging from this ribosome. There's a really cool animation that shows this, and I'm going to pop this up. This really does a great summary of this whole thing we're talking about here, the central dogma.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Well, there you have it. That animation does such a great job summarizing a very complicated set of events in just a couple of minutes. So I highly recommend you give that a few watches. It really helped to illustrate this whole process we're talking about and make it more familiar to you. Okay, so we're going to move on and practice this a little bit. This is the type of thing I could do on an exam. I could give you the genetic code. I could give you a sequence, a set of bases on an mRNA transcript, and you would have to then use the code to determine what the protein would be that is built from it. Okay, so on the transcript I have 3 base triplets already sort of determined for you. That might not be the case. It might just be a sequence of letters, and you have to sort of establish what's known as the reading frame, figuring out what the codons are. To do that, you always look for AUG. AUG would be your start codon. Okay, so you can go ahead and pause the video and work through this, and then I'll go through the answers here in just a minute. Okay, so the answers would be the following. You have AUG, that's methionine, that's your start codon, so that sets the reading frame. As soon as I see AUG, I know that the next 3 units are the codons, okay? And so they would be looking like this. So the next codon is GAU, so GAU, which is this amino acid here. Okay, the next one is CGC, which would be CGC, our good friend arginine here. Then it's GUA, valine. Okay, and then it's UAU, tyrosine. And then UGA tryptophan. Oh, sorry, UGA, which is our stop codon. Okay, so that would be stop codon. So that tells you that translation is complete. So it would be, this would be the polypeptide that would result if this particular mRNA was read through a ribosome and translation were allowed to occur. Could you do something like that on an exam? It's just as simple as figuring out where your start codon is, setting the reading frame, figuring out what your triplet codons are, and then using the code to determine the sequence. Okay, from here we're going to talk about mutations, and this is a good place to talk about them. You know, they often happen during DNA replication, which is something we talked about earlier, but to understand their significance of what they do and how catastrophic they can be, it's sort of related to what we're talking about here with gene expression. So we'll talk about mutations here. Now, keep in mind here our transcript and our genetic code. Bearing those in mind, these are the different types of mutations that can happen. You can have insertions, deletions, and substitutions. And just as the name suggests here, an insertion would be if a base was added into the gene. A deletion would be if one was removed from the gene, and a substitution would be if one of the letters was substituted with another. I used to study Duchenne muscular dystrophy, and we used an animal mouse model called the MDX mouse. And the MDX mouse has a substitution in it where there's a C that is converted into a T, I believe. And that one mutation has a very catastrophic effect because it makes the protein dystrophin, it makes the normal protein dystrophin not produced. So MDX mice do not have a full dystrophin protein. Okay, now the effect can vary. So you can have a variety of different effects that result from these mutations. If the mutation only affects one letter, one base, it's called a point mutation. But that point mutation can vary in its significance or its how catastrophic it is. So a silent mutation is one where there's something that happens to the base. It's most often a substitution. One of the bases is substituted for something else, but it has no consequence. It's silent. And to understand how this could be, take a look at this genetic code. What you're going to notice here is that for a lot of these, the third base in the codon is not really all that significant. They sometimes call it the wobble base. And so in any box here where one amino acid can be coded by any of the four third letters, that would be a case where you could have, if it's the third letter in a codon where the mutation occurs, so like let's say, looking at this picture, let's say here's an AUG right here. And so let's say that the mutation happened earlier on in the S phase when the DNA was being replicated. And let's say in that particular cell, this G was not a G, but it was instead like a U or something. Well, because it affects the third letter in the codon, it might not even matter because all different letters might code for the same amino acid. And that's the case for a variety of these, like for proline, for leucine, for threonine, for alanine, for valine. Sometimes it will matter, the third letter, like here, you can have an AAA code for this one and an AAG code for this one, but an AAC code for something entirely different, right? But for a variety of these, the third letter doesn't even matter. So that would be called a silent mutation. The mutation has no effect. The same amino acid will be added even if the base is altered. The DNA template that built the transcript, if that base is altered and the transcript still transcribes and translates the same protein, no big deal. That's a silent mutation. Okay, a missense mutation is one in which the mutation leads to a substitution in the amino acid. And generally it's just one amino acid. So let's say, let's say this transcript codes for phenylalanine, serine, tyrosine, arginine, serine, and then just one of those, if the mutation happened, let's say in this particular base, and that caused this to not be a G, and so that it resulted in just only one of those amino acids being swip swapped with a different one, that would be called a missense mutation. And these can vary in how significant they are. It could be the case that in a big long protein, maybe several hundred or thousands of amino acids long, it might be the case that just having one amino acid wrong doesn't actually have any effect really. Or it could be the case that the particular amino acid that is substituted is a very important amino acid in the protein, and that just just having one amino acid off might greatly alter the protein's function. It all just depends on the situation. It depends on the chemistry of the domain. It depends on where in the protein is and what's being substituted for it. It can all vary. Okay, a nonsense mutation would be one where something happens to the DNA and it results in a premature stop codon. Okay, so that would be like, the various stop codons we have are UAA, UAG, UGA. So that would be, sorry, got my baby here. So I'm trying to think here where that I would have one of those. Okay, so UAA, UAG. Do I see a UAG anywhere? UAG. What about a UGA? Do I have a UGA? Okay, so we'll just say I should have planned this out better before I recorded this. Okay, so AA. Here's one. Well, I'm not going to try and figure it out. But in essence, what that would mean is if one of these codons, which was not a UAA, UAG, or UJA, so it wasn't normally a stop codon, but there's some mutation in the template strand that causes there to be a premature stop codon in the transcript, that could be majorly catastrophic, because what that does is it signals the end of translation. It could be that the protein only gets a couple amino acids added to it before it's terminated, or maybe the protein almost gets complete, but then it's just terminated just prior to the completion of translation. So a nonsense mutation does have the potential to be pretty catastrophic, depending on how upstream or downstream it occurs. If the nonsense mutation occurs right away, way upstream, then everything downstream of the transcript will be cut off. So like, in other words, if this RNA polymerase was going to keep going and this mRNA transcript was going to be like 1,000 or 2,000 bases long, if the stop codon gets put here and not 2,000 bases down, this whole thing is not going to code for anything. The whole entire transcript will not code for anything. It will just be garbage. So that would result in a major change in the protein. Okay, a frame shift mutation is one where it's usually when there's an insertion or a deletion. We don't really get frame shift effects here if there's a substitution. But if a base is added or taken away, what that can do is shift the reading frame. So right now my reading frame looks like this. I have the AUG, that sort of sets the reading frame. And so here's my codons. Well, if one of these got deleted, let's say, then all of these bases shift over. And now every codon downstream from that is going to be different. So that one small insertion or deletion might have a huge effect, because now the entire reading frame is skewed and all the proteins that are added could be incorrect downstream of that. Okay, so a little bit about mutations, and the severity of their effect really just determines, it's really situation specific. Silent, no effect. Missense, one amino acid is replaced with another. It can or cannot be problematic. Nonsense, there's a premature stop codon. Again, depending on how far upstream or downstream it is in the gene, it could be catastrophic. And then frame shifts are usually pretty catastrophic. it would be rare to have a frame shift mutation where the whole reading frame is shifted and it doesn't have a major effect. Okay, so that's a little bit about mutations and how they fit into this whole scheme of the central dogma of biology.